and welcome to a quick tutorial on measuring membrane proteins with mass photometry. My name is Austin Graves and I'm a senior field application scientist with Refeyn. Now, a common question we get from new users is, can I use mass photometry to measure samples in detergent like membrane proteins? We talk a lot about how detergents create background noise in our sample, and this is what makes analyzing membrane proteins so tricky. Of course, it's easiest to analyze membrane proteins with mass photometry that are prepared with other mimetics like nanodiscs, peptidiscs, or SMALPs, but let's say that detergent is all you have to work with. Well, in this video, we're going to look at some membrane protein data, and I'm going to give you some tips on how to get the most out of this kind of sample. All right, let's go ahead and jump into Discover MP. I've got a workspace open here with a sample of clean PBS, undiluted LMNG buffer diluted 10-fold in PBS, 100-fold in PBS, and finally a membrane protein sample in LMNG buffer that was pre-diluted in PBS. Before running our protein, it's always best to look at different dilutions of just the detergent buffer so we can determine how much we need to dilute the detergent to achieve an acceptable level of background noise. One thing you should keep in mind when doing this is that the acceptable level of noise is going to depend on the size of the protein. You can imagine that a larger protein complex is going to be easier to distinguish from background signal than a smaller one. Now, anytime we run a sample, it's not unusual to see some background signal from our buffer. This mostly is going to appear as two mirror peaks on either side of zero like this. This movie of PBS shows the minimal background signal we can expect when running a clean buffer. However, high concentrations of detergent, in this case LMNG, produce a substantial background signal. You can see this reflected in the messy signal in the movie, as well as the large mirror peaks in the histogram. In this case, we're seeing a peak of about 170 kilodaltons, so you can imagine if we're working with a protein that's smaller than this, we would not be able to distinguish it from the background noise. Now, if we dilute the LMNG buffer just 10-fold in PBS, we can still see some background signal, but the movie and the data have cleared up considerably. Now, once again, how much signal we can tolerate is going to depend on the size of the protein. So if we were looking at something that was 60 kilodaltons or smaller, this still wouldn't be dilute enough. So if we go down to 100-fold dilution, we can reduce the background signal even further. The protein we're looking at in this particular experiment happens to be over 100 kilodaltons, so a background peak at 59 kD, like this, would be perfectly acceptable. Now, the way that you're going to want to go about diluting these samples before running them is to have your protein at high concentration in its native buffer. Work quickly to make appropriate dilutions and then measure it. That way you're able to get representative data before your sample deteriorates. So that's what we've done here. So in this sample, what we find is we see a peak at around 50 kilodaltons. That's probably going to be the noise coming from the remaining detergent, but we have a peak of our protein at an expected size of about 114 kilodaltons. We can also see some larger species in the sample, which could be aggregates or other impurities. Also, when we look at the movie, we can see that these clear point spread functions apart from the background signal generated by the residual LMNG. So I hope this demonstrates that with appropriate planning, controls, and a little bit of finesse, you can certainly measure membrane protein samples with mass photometry. Thanks for watching, and if you'd like to read more about running samples with detergents, please check our app note, which is linked in the description. Thank you.